Greetings, everyone. This is the NOAA EBFM EBM seminar series, second Wednesday of each month, 3 p.m. in the evening, noon here. We're actually in the Portland, Oregon office, hosting it here. Thanks, West Coast Regional Office, for hosting us today. Our speaker is Dr. Brian Wells from our Southwest Fishery Science Center, who will be speaking about process studies to quantify ecosystem dynamics and inform EBFM in the Central California current. Brian will start his presentation, and Judith Salter in our NOAA Library office is fielding questions at the end of his presentation, so feel free to type in any questions you may have as we go along here, and Judith will do a great job of fielding those questions. So Judith, anything else we need to announce at this hour?
Well, thank you all for joining us, and we'll get started. Thank you, Peg.
Okay. All right. Brian, you're on.
Thank you all for attending and being interested in the work, and thanks to the regional office for housing us. What I'm going to try to accomplish today is walk through about a decade of work that our group in Santa Cruz has done, and try to place it in the framework of how we think we should move toward ecosystem-based management. So what we're going to do is try and frame our work relative to the roadmap. And for clarity, how I'm interpreting this roadmap is these 6 steps that we've all seen at this point, I would guess. With step 1 being, what are our objectives? And this is the fisheries ecosystem plan level. This is the trying to understand generally what is known about any system that you would like to manage. And then that second level, which we're going to devote a lot of time in the weeds on, and then we'll come back out of the weeds and go 30,000 foot afterward, but what is the fundamental science we need to really build out our understanding of a system? What are the processes that need to be understood, and how can those then be used to move to step 3, prioritizing risk in the ecosystem? And then, of course, we get to the fun part of what are our trade-offs, what should we be thinking about in the ecosystem, and then providing management advice. And then we'll go down a little bit of a soapbox path for number 6 of what do we think some of the things we need to consider to maintain a resilient ecosystem in the Central California system. So starting with that first layer, what are our objectives? At the beginning of any of these studies, whenever a program is developing to try and be applicable to ecosystem-based management, it's clear that you need to develop some sort of concept or conceptual model of the system. Ask the people that know, the literature, the research, whatever the current understanding is. And these can look a lot of different ways, from what we've used there in the upper left to inform the IEA, the California Current IEA, to what has been developed here in the Northeast, which is more of a what are the connections and the significance and type of connections. But regardless of how these look, their main goal is really to frame our questions. After a decade, we've come up with this conceptual model for salmon recruitment. And a lot of this talk is going to focus on salmon, but don't make a mistake about it. We're really focusing on the things that eat salmon, things salmon need to eat, and the environment that's determining salmon success. But after about a decade of work, we've been able to design this conceptual model, where we actually go from the physics in the winter in the upper left to the spring on the bottom, and all the way down till we get to primary production, forage, Chinook salmon, predation, and so forth. And each one of these arrows, in effect, was a question we had over that time period, and we'll dig into some of these arrows specifically. But the primary point here is, if you've got a conceptual model that is, generally speaking, representing the system, that's excellent. If you have a conceptual model that you can actually derive the mechanisms and processes, that may be more stable in a changing environment. So what is the fundamental science you need before moving forward with actually applying your work to ecosystem-based management? Again, salmon is the focus in this case. I and members of the Northwest are actually designing the next decade of research for salmon along the West Coast. And what we've done is we've taken that conceptual modeling approach, with, in this case, the blue at the top being environment, and then whittling through these different arrows if you wanted to. You can look at that later. But the real point here is that this is now our starting point for designing this research program. We went out, we asked the literature, we asked ourselves, we asked everything, what connects to what and what's important. But the critical part of this is you'll see in this conceptual model not only the boxes, which ultimately indicate, or ultimately are attributes or indicators, but it's those arrows. And our questions revolve around all those arrows. They define our actual research foci. So now, as we move forward with the salmon ocean ecology, or our general ocean ecology program, we can pigeonhole or at least focus people's questions to address these errors, so when we're done, we've got a relatively fully mechanistic understanding of the system, which we can then apply to trade-off analysis and management, as well as risk analysis. Now, I'm going to get in the weeds here for probably 10, 15 minutes. But in order to fill out that type of conceptual model, we argue you have to take an ecosystem oceanographic approach. And this is defined as Curry et al. 2008 defined it. Specifically, how does environmental variability or secular change affect the trophic structure? Who eats who is there, who is not under different environmental conditions? So no longer are we thinking of salmon as the only critter that we're interested in managing, but in fact, they're food and they're predators. Now, we're fortunate in Central California. A body of research tells us we can tell a lot about this system with these 5 critters that are on the screen. Specifically, to the right are anchovy, juvenile rockfish, and krill. And these 3 forage members actually vary in some known way to the environment. And in fact, you get, as an example, these adult northern anchovy coming into our shelf when the conditions are poor, and we'll dig into this more. Whereas you get juvenile rockfish and krill when conditions are good. When salmon come out, they rely on these forage members to actually make it to the next stage of their life. But they won't make it to that next stage if common murre, a piscivorous seabird in the area, feeds on them. And we'll see the switching trophic relationships that can be caused by environmental variability that ultimately result in common murre having a significant impact on Chinook salmon. Common murre in our case is also basically varies with many of the predators, so it alone is not the evildoer in this story. It's just representing predation in the area. So if we remember, the first conceptual model I showed you of salmon started on the left with basin scale drivers, and all the way on the right, we had salmon survival. So our first question set out over a decade ago was simply, can we link, just with no mechanistic understanding, these basin scale drivers to some sort of salmon dynamics along the entire coast, actually. What I'm showing you here is the strength of the North Pacific high relative to the Aleutian low ultimately controls the strength of the California current. And in this case, it's strong. You get a fat green arrow. Now, with that arrow, we could assert that the mechanisms of increased upwelling, increased transport, but by whatever means that we'll uncover, we see that the salmon, shown coho with a little c and an arrow, ocean-type Chinook and stream-type Chinook, somewhat outdated terms, grow more. They come back bigger to the California current when the California current is faster. So right out of the gate, we found that, yes, the environment in some way is determining salmon size dynamics coming back. But from an ecosystem management standpoint, that correlation can be somewhat unsatisfactory. So we then dug into what are the connections between that North Pacific high and ultimately the survival of a salmon. Isaac Schroeder provides a really good step forward in trying to understand how these basin signals relate. What I'm showing you in the map is the area of interest in the box, that light blue. Now, it might seem myopic that we're sort of going to pay a lot of attention to this area, but in fact, it is the most biodiverse area for mammals and seabirds. The largest seabird populations are there. One of the largest Chinook salmon populations come out. And it's an enormous forage community. So in effect, not only does it correlate with the California current dynamics, it actually acts in some regards as a bellwether. So that said, we're going to focus on that area. The circles at sea show the average location of the North Pacific high pressure cell through the 12 months of the year. You can see it waxes and wanes, goes farther off and comes back. The color of it, red being stronger, represents its size. You can see its size changes over time as well. And these are the average conditions, where it is on average in each month, the strength of it each month. But look at the little figure inset there. That's the amount of variability across those years you actually get for the size and location of that North Pacific high. It varies dramatically in space, and it varies dramatically in terms of strength. And Isaac got the idea with collaborators Bill Seidemann and Brian Black and others that in fact that March location of the North Pacific High should in some way relate to production on the shelf the next spring. The argument being that a larger North Pacific High, a closer North Pacific High in March will allow for an earlier initiation and availability of nutrients on the shelf. And in fact, the figure on the right shows just that. He develops a preconditioning index with the right number being a larger, closer value for the North Pacific High. And then the y-axis represents productivity of seabirds, rockfish, and a variety of other critters. And what we see, in fact, is that the stronger the North Pacific High is in March, the greater the community productivity in late spring. So in essence, what we see in the spring and summer when seabirds are reproducing, when salmon are coming out, the productivity in late spring is related to that March condition. Also note, from an indicator development standpoint, that is a nonlinear relationship. So it could, in effect, provide a threshold value if one is interested. Let's dig a little deeper. That top figure shows a time series of krill in the gray for 1990 to 2010, collected on the shelf in June. The bottom left figure, you'll notice a lot of very small dots here. We've taken regional oceanographic models and developed an index of upwelling in each one of those dots, upwelling and transport actually. It's the isopycnal depth, D26.0 for those that are interested. But the take home is, using the ROMS data at each one of these points, these little itty bitty points, we calculated a value representing upwelling and transport in March, reminding ourselves that that large basin scale indicator some way is playing out. We then correlate this gray time series of krill to every one of those locations, and we find that krill on the shelf in June is highly correlated to the upwelling and transport dynamics in March, right in that location. So in effect, then it continues to be correlated up through June. So in effect, we argue that nutrient availability in March ultimately relates to the number of krill on the shelf in June. And looking back at the literature, it makes full sense. Don Kroll, Garcia Reyes found that approximately 100 days following the initiation of upwelling, which in this case is March, you find krill developing. So it fits our idea of what's going on. Another thing to look at is this other time series. In fact, gray was rockfish and black was krill, sorry. The gray time series is the rockfish now, juvenile rockfish, and how they correlate to March conditions. In other words, how many juvenile rockfish are on the shelf, a very important forage item, in June? How does it relate to March conditions? It's worth stepping back and sort of reviewing how juvenile rockfish come to be on the shelf in general. They're born on the shelf break as passive larvae, and they're born in March and February. They then end up on the shelf by some sort of transport, and in June, when seabirds are reproducing and salmon are coming out, they're either available or they are not. And what's really cool is the only month that was correlated to upwelling values at sea, again, these blue dots, these little dots, ROMS data was calculated for each one, and then that time series was correlated to it. We find that rockfish on the shelf in June, these black dots, are correlated most strongly to upwelling and transport dynamics right on that shelf break. So for review, the size and location of the North Pacific High in March relates to the abundance of krill on the shelf in June due to early introduction of nutrients. Most likely, rockfish end up on the shelf in June because of initiation of transport in March brought on by the North Pacific High. Now what happens if you do not have strong transport or upwelling in March? What if it's a poor condition? Well, we can look at it in this way. The top figure shows krill collected at each one of those dots shown here we then correlated the time series of krill to the oceanographic condition. And it turns out you get a lot more krill in the area, specifically in the Gulf of the Farallones, which is this location, when you have subarctic conditions. In other words, when you have upwelling. And we kind of just showed that. But when you have upwelling, you get more krill, specifically in the shelf. Rockfish are similarly correlated to krill, which is no surprise, except more distributed everywhere. The key here is that, as we just showed in the previous figure, you get more krill and rockfish under subarctic or cool upwelling conditions. But equally important to our story is anchovy. Those krill and rockfish were born the same year. But what happens is if you get poor upwelling conditions, referred to here as subtropical, you get more anchovy instead of juvenile rockfish and krill. The opposite color just simply says it's correlated in the opposite direction. So now we've got a dichotomy. If conditions driven by the North Pacific High represent great conditions, we get krill and rockfish on the shelf in June. If they are not great conditions, we get anchovy. And this will be important as we start telling our story. It might seem a little overly specific, but it actually is also important. During subarctic conditions, as I said, you get rockfish and krill. Those rockfish are distributed in areas such as this, shown by the blue, rockfish, and here. When you don't have many rockfish and you get anchovy, they're distributed just south and very close to shore. And this will become a very important aspect of our story when we talk about predation and forage switching by seabirds. So that's kind of the weeds of how the ecosystem sets up. The North Pacific High in March, depending on how big it is and close to the shore it is, you get a certain amount of nutrient introduction early in spring or late winter and transport that's appropriate to bring krill or rockfish to the area. If it's not strong enough, you get anchovy in the area. Rockfish and krill distribute pretty widely, anchovy mostly in the south and coastally. Now what's really neat, I don't have any particular yen for salmon, but they really offer a very nice experimental unit. They haven't been in the ocean during all of this, but they go out in June and have to experience it. So in an ecosystem context, we can really ask a lot of questions using salmon. As an example, the first question is, does it matter when you go out? Remember, if March conditions are good, June you'll have more krill and rockfish. So what Will's done here is he's looked at different times of release for salmon from hatcheries in the Sacramento River area in Central California. I'll start by saying you can ignore this. It's early in the time series and represents very large fish released very late. With that in mind, what I'm showing you at the bottom is the time since upwelling began. Another way to think of this is somewhere in March or something, and then how many days after that is best for a fish to go into the ocean. And then predicted survival here. And the key part is this hump right here. In fact, we find that mortality or survival can vary 40-fold in this earliest period at sea. And what we find is that if a salmon comes out approximately 90 to 100 days following the initiation of upwelling, which typically occurs in March, has the greatest survival. And we find that that specifically relates to the development of krill on the shelf in June. Reminding ourselves, krill develops approximately 90 to 100 days following the initiation of upwelling. Specifically, krill in that area B. Turns out if salmon come out and they eat krill, the greater the portion of their diet is krill, the greater their condition. So in effect, if they come out at the right time and there's the right amount of upwelling, they'll eat krill and they'll be fat. Now this is really an area that I think is really becoming exciting for us in the next few years. And it's going that next step of Curry et al.'s paper and a variety of others since. It's not good enough just to go out and do empirical studies. We go out at a specific time, a specific location. But in fact, if we can pair those empirical studies with modeling approaches, we not only can build ecosystem models that can be used to test trade-offs, but actually can use those models in effect to corroborate what we're finding in the empirical world, as well as develop new hypotheses. Long story short, the top left figure shows the survival of simulated salmon at sea during years which we know were very good survival. Basically, it's a ROMS model with krill being developed in it. Salmon particles use a restricted search pattern and feed on krill that they can get to. And then using a dynamic energy budget model, they grow. The key take-homes of that upper figure are that growth is great, as indicated by red, and they distribute widely. During years of poor survival, however, growth is poor, and they're very close to shore. So that's a nice spatial temporal idea of what we can expect to see in the system, and goes beyond what we could see on the ships. What's also neat, we can go back in time and we can look at the environmental conditions that ultimately resulted in these good and poor survival years. That top figure shows the different months of the year, with blue being the time series for temperature of the years where survival was good, red being years where survival was poor. Notice these good survival years are always associated with the initiation of upwelling in March, as indicated by cool temperatures. Following, phytoplankton blooms are better during the years of good survival in the blue. And in line, May, June, you start getting these bumps in zooplankton. So in effect, the marrying of an empirical study with a modeling study has allowed us to open up the gate for many more questions. We've taken this study even farther, and now what I'm showing you is, in the top figure, our estimated survival from coded wire tag analysis, and then the estimated survival from that model using growth. So we can estimate the survival of these salmon really, really well just by looking at their growth and distribution patterns at sea. Same data shown in a different way. Blue is the VPA, and red is our estimate from the model you just saw of survival. Why does bigger matter? Well, now we're getting into things that eat salmon. This star represents the Southeast Faroe Islands. And on that island is the largest breeding population of common murre, piscivorous central place feeder. The murre come off of that island in May and June and I think July to get forage fish to bring back to that island to feed to their young. We have both at-sea observations of where they go, and we have island observations of what they bring back. The question is, do they change their behavior under different environmental conditions that lead to different forage communities? That top figure shows just how fast or how big the growth is of this population, even since 1983. There was egging following the gold rush, and not until protections were put in, and even after that a bit, we started seeing a spike in the population from what was 36,000 in this case to 250,000, just on the Gulf of the Faroes, shown in that black dot. At sea, we observed the densities or distribution of these birds grabbing food to take back to their young. And we see during productive years, reminding ourselves March conditions were great, there's a lot of krill, there's a lot of rockfish. These birds feed everywhere. There's no hot spots. Their average foraging distance is about 90 minutes. When there's low production, birds either fall off the island or, with other colonies of the like, can feed inshore in higher amounts, right where we know there to be anchovy. We're so lucky to have this bird. The top figure shows what they eat. The blue is the amount of rockfish they eat over that time period, reminding ourselves they bring it back to the island when we look at it. 90% rockfish during those productive conditions. When they're not eating rockfish, they're eating anchovy. Again, 90%. So it's one or the other with this seabird. But reminding ourselves, when they're not feeding everywhere on rockfish, they're coming inshore where anchovy are. And reminding ourselves, when the conditions aren't great, the salmon are inshore where the anchovy are. When anchovy in the diet goes up for these seabirds, salmon in the diet goes up as well. Now, it's only 8–10%. But back-of-the-envelope calculations tell us that this can be substantial. Bioenergetic demand of even this incidental predation can be significant. And it is. What we've done is we've now taken survival of salmon and related it to the various aspects of the ocean system and freshwater system. The top figure, we have to take care of freshwater environments after all, shows the relationship between survival and April discharge. You get higher survival of these critters, these salmon, when the discharge is greater. It allows them to get to sea quicker before getting preyed on. It also allows for a larger habitat so they can grow better. The second figure here shows survival related to that March upwelling, and in fact it's parabolic, which is neat because that tells us there's an optimal window. If we wanted to dig in even farther, I could show you results where if there's too much upwelling, you get advection of forage and chlorophyll from the shore. The bottom figure is the one that is most interesting to me, though, and it shows the interaction between the availability of rockfish on the shelf and the abundance of common myrrh on survival. Red is low survival, blue is high survival. And if you look at this x-axis with rockfish, if rockfish are -2 standard deviations below their normal, the survival of salmon is not really very much affected at these low levels of common myrrh. White is medium. So even at the very lowest levels of rockfish, common myrrh are doing okay. The salmon are doing okay in terms of their survival. But once you start pumping common myrrh into the story, and once we realize what we've talked about, that they switch from rockfish to anchovy and incidentally prey on salmon, the mortality on salmon is exceptionally high. So this provides us now, contextualized from the basin scale, all the way to whether salmon is going to be eaten, how the environment affects salmon going to sea. So now we have this understanding of the system. We know what's important, we know what's less important. But we're often asked on a year-to-year basis, what are the risks, what are the concerns coming up? Now we can take a relatively simple red light, green light approach, and we can extend it somewhat by adding it on to our now mechanistic understanding of the system. And we can provide information to the PFMC. For instance, in 2015, everything's red. Turns out in 2017, as these fish return, no surprise, there are very low numbers. And this is a very basic way of providing an understanding of what may be coming down the system. But it's unfairly simplified. In fact, I think we all understand these aren't just linear effects of one box determining the next, and then you can add it up and say, here's what's going to happen to salmon, things salmon rely on, seabirds and the like. But rather, it's an integrated risk. And we really saw that when we looked at how the myrrh feed on one thing, groundfish, which is also managed, or feed on another thing, anchovy, which is also managed, and when feeding on anchovy, feed on salmon, which is also managed, and the amount of salmon out there is going to be dependent on the freshwater flow. So there's a lot of things that can be managed in just the simple model I've shown you. So when you consider those interactions, this may be a more important thing to show to managers. In fact, these nonlinear thresholds, these interactions that occur. So sure, I've been talking about salmon. These are managed, and this is managed. So as these predator populations increase, as we start recognizing their impacts on salmon and forage communities, and vice versa, back and forth, salmon competing with the predators as well, we need to start thinking about the trade-offs and providing, when we have a system, in this case I'm going to focus a little bit on the predators, when we have a system where most of the predator populations are going up and up and up, we need to start taking these process studies that we understand and bringing them into some kind of framework where we can really talk about the trade-offs that increasing predator populations can have in the face of needing to manage the forage and the salmon and freshwater flows. A recent study by Brendan Chasko out of the Northwest really puts this in a good framework. What you're seeing here for primary mammal predators along the entire CCLME, you see the biomass of juveniles they can eat, the numbers they can eat, and adults. And we didn't talk much about adults here, but there's an impact there too. And the key is that as each one of these predators increases, the bioenergetic demand also increases. So how do we manage in a world where not only are we having to compete with environmental variability, freshwater needs, forage availability, but also predators? And we can go back to some of our modeling efforts. This figure shows a model by Jerome Fisher, where, without getting into specifics, on the left you can look at sea lion abundance and distribution, and he's modeled this behavior of sea lions relative to the abundance and distribution of sardine, a primary prey. So this type of model allows us to actually understand how do predators relate to their forage community. We're in the process now of bringing that salmon IBM into this, so we can overlay this on top with salmon, and we're building out the forage components by talking about the rockfish coming into the shelf and the seabirds. So this model here, in addition to the salmon model I showed earlier, are being brought together so that whenever we try to understand mitigation efforts or trade-offs, we can do it in the context of populations we're trying to recover. When we go and we try to give advice, there's many ways to do it. And we're coming back out of the weeds now. And one of the ways to give advice is to do what I just did. And that's a bit more than most people need when they're trying to manage a population. Another way to look at it is we can provide a level of complexity of a model where the more complex that model gets, sure, the bias comes down, but the uncertainty goes up. And we've battled with managers, specifically tactical management. And I've been on papers for salmon demonstrating this very point. And when you start bringing in these environmental variables or preyscapes or what have you, the uncertainty can get great. And while it's encouraging to see high R squares, if you keep throwing these variables at it, you can end up with a model that's far too complex and ultimately fails due to one link, especially if that model is not based in some way on mechanism. But super simple models suffer from missing the larger story. And that's where a sweet spot can be found. Another way of wording that is the model needs to be built to address the question at hand. It needs not be more complicated than need be, but also needs to be complicated enough to address that question. One approach we're taking is a life cycle model for salmon and all the different things salmon run into in their lives, from egg to return. The top figure in the black bars shows the abundance of salmon returning to the river. And you'll notice it's highly variable from some of the highest we've seen in that record to a fishery closure. The blue bars represent our ability to model that number of salmon coming back to the rivers. And this has an R square of 70%. And cross-validation shows its quality as well. But what's neat about this model is you can actually provide a sensitivity of that population to various things it experiences in its life, and therefore provide advice for what areas might be of issue and areas that might need to be tuned for proper strategic management of the population. Being clear, not tactical management. I'm not saying we're trying to predict how many salmon are coming back next year, but rather, given different management scenarios, how do we expect this variability in the population to change? How often would we expect closures to occur? So we take each one of the variables and put them together into one model. And there's that model in dark blue at the bottom. And we get that nice high R square. But we can actually evaluate sensitivity by selectively pulling out individual covariates. For example, if we take out carrying capacity. And this model's in development, so don't take these values home, please. But we can take carrying capacity out of that model, and here's how good the R square is in the light blue with carrying capacity out. Here in the gray is how good that model is with only carrying capacity. North Pacific gyre, a correlate to North Pacific high. Temperature during incubation, flow during outmigration. Notice it's one of the most important variables, reminding us of our model with April flow. Gray is very big. You can model a lot of variability just with flow. And then predation, being the highest influence of the shape of that returning number. And we know why all these things work, and we can explain it. But now we can say, these are the things to which your population is most sensitive. If you were to turn handles of flow and temperature, you may have some control on this variability. As a sideline, flow and temperature also may make the salmon more robust to predation at sea. That mechanism is not fully fleshed out. So to this point, what I've said is, you need to set up your research program out front to understand how it can be used for ecosystem-based management. Identify the questions. Build out a mechanistic understanding when possible and well-funded. And then you can start going back to trade-off models that are actually built with this understanding of mechanism. Provide an appropriate model for the question at hand, and then you can provide advice in terms of trade-offs associated with different levers that you have in your control. But the state of today may not be the state of tomorrow. And to maintain a resilient ecosystem, our top level, you've got to be willing to revisit that conceptual model, revisit those arrows, and ask, how do those arrows vary under different secular changes in the environment or community that you have? For example, that upper left figure from Sideman's group, Fairlands Institute, demonstrates that the amount of environmental variability in our system has been and may continue to be increasing. And I built out the story of how environment influences forage abundance and distribution, foraging behaviors of predators, salmon survival. Swinging back and forth more and more as the environmental variability increases. And swinging even greater. And I'm not trying to pick on common murre, we're all increasing. And we need to consider how we're going to manage under the scenario of increasing environmental variability and increasing predator populations. This little cartoon has to make it in all talks now because I love it. And basically just lays it out. Under suboptimal upland conditions in March, you get fewer preferred prey in the area, more anchovy, or predators come inshore, and this includes sea lions, possible humpback, a number of predators, and when inshore can impact not just their alternative prey of anchovy, but the salmon that interact with that. We need to think of how we're going to deal with this going into the future. And those modeling efforts, like I showed you of Jerome Fleischer, in combination with our parameterization from empirical data, is an extremely valuable tool for estimating what the future can look like. And to give you an idea of the kind of complications, and as Curry put it, ecological surprises that we've had, is work that's headed by Jared Santora at UCSC right now. We saw an unprecedented number of whale entanglements with crab gear recently. And it wasn't until you actually unfold the story and unfold a little bit of the mechanism you can see why. We had a large marine heat wave that led to fewer krill on the shelf break. In addition, we also had a domoic acid outbreak, which made the pot fishery occur later, more condensed, higher density for a shorter period of time later in the season. Because there were fewer krill, whales moved inshore, and that combination turned into a lot of entanglements. We can't envision these types of ecological surprises, and we're going to have trouble managing at an ecosystem level with such ecological surprises. But having a mechanistic understanding with retrospective data and building out models that take us into the future and allow us to play with the world may help elucidate these types of surprises. And that's pretty much how we've contorted our research to fit into these steps. not very limber, but I think we may have accomplished it. The primary point being, at this point, when we start developing new programs or have programs that are in operation, we should revisit how they interact, each one of us in each one of the studies. Develop a research program that leverages those efforts and places our research in a way that can actually be used to inform and better accommodate a need to explore trade-offs and provide advice. And I've talked as much as I can. I'll turn it back to Peg. Thank you very much.
Thank you, Brian. Excellent. I really appreciate that detailed analysis. Judith, what I thought I'd do is maybe turn to your location now and see if there are folks in the room that might have questions. We have a few folks here, but we'll start maybe in Silver Spring. So folks have any questions for Brian?
Came from an hour talking about the situation with southern resident killer whales, and so just the kind of the swirling together of the ocean conditions and the way the predators are moving to chase the prey, and just the effects that are so profound. For example, with now sea lions moving in and competing with the southern resident killer whales for the Chinook prey basin. I have just kind of spinning right now with regard to what to do with this information and apply it to kind of these urgent situations, such as what we have with southern resident killer whales and the whale entanglement example that you just gave.
I think it goes back to what I'm trying to impart is that all of the stuff I've gone through is necessary to underlie appropriate models. But the customer-facing side of this effort should be packaged models, so to speak, that allow us to just ask those questions. And when done properly, we shouldn't worry about whether a mammal comes inshore, offshore, there's more or less fresh water, for example, but rather trust that it's parameterized well with studies like this, and then just ask those models the questions we need. What I've tried to do here is less make a brain swirl about what's involved in it, and more try to gain confidence in it, to understand it from a review standpoint, but then step back and say a combination of these process studies can feed into these management models that allow us then to just ask those questions.
And it did help build my confidence, and then I guess the question becomes, are we heading into a situation where there can be some agility with regard to informing decisions about, well, we project there's going to be poor salmon production in an area where southern resident killer whales are going to be, and does that inform how we want to do hatchery management or habitat, targeted habitat?
Sure. In the example you're asking specifically, and I didn't get into it here, but we do have distribution studies for adult salmon. An understanding of how they distribute differently under different environmental conditions allows you to know where they may intersect killer whales. And knowing where they may intersect killer whales allows you to change spatially harvest control rules year to year. So that's how I would use it in that regard. I think that more generally speaking, if the question were about salmon, let's say, I'd say focus on freshwater and focus on trying to save anchovies for things to eat so they don't eat the salmon when they're trying to eat anchovy. The questions would be, how do we provide, you have brought up hatchery fish. How do we provide the food that killer whales need? Maybe we need to actually evaluate, are hatchery fish helping or hurting salmon and seabirds? And are they helping and hurting killer whales? All in one framework. So I see it as helping tactically if the specific question is well formulated and modeled. Generally speaking, I see these approaches as more strategic. Like, what does a hatchery program over the 20 years look like relative to an increasing demand of killer whales when the environment's changing also? Rather than, this year let's not fish over there because that's where the killer whales. I'm not managing.
So, Judith, we're turning back to you. Any other questions?
Thank you, Peg. We have a question from Michael.
Dr. Wells, good afternoon. My name is Mike Dirks. I work with several Bay Area utilities and Northern California utilities as a regional liaison with the Water Research Foundation. And every once in a while, there's considerations with drought and low reservoir levels and the temperature for salmon across the region. And I was curious with your study if you had any particular advice for water resource managers, reservoir operators, and water utilities about what they can do to communicate about the sensitivities around the habitat for salmon, and how to better manage the inland resources for the smelt before they return back to the sea and the salmon that spawn. I'm just, this is probably beginning a conversation to better understand the issue across the region.
I'm not the best person to ask that question to, but I can answer some degrees of that. We have found in size-selective studies that smaller salmon during poor oceanographic conditions, as I've laid out here, die first through predation. We have found that if the salmon had grown better in fresh water, which can be affiliated with temperature and flow, that salmon is more likely to survive in poor oceanographic conditions. The problem with that rather simplified answer, however, is how often are there poor environmental conditions? And if you were to just promote bigger, and I don't know the answer to this, to be clear, but if you were to promote bigger, more salmon going out by increasing or doing appropriate freshwater management, you may raise the bar in terms of the average number of salmon coming back over time. But without understanding the competitive forces between more, larger salmon and the interactions they may have with hatchery fish, you'd be making an ill-informed decision in my mind. I think the global answer to this question would be that the appropriate way to answer that is to continue building out these models, and we're not too far from that answer actually, to say what would different scenarios provide. The other global answer to this question is, what is it you're looking for? I think that we often, and I don't know the answer to this either, mind you, but I think we're often saying, oh, we just want more salmon back, raise the bar, more salmon, more salmon. But in fact, perhaps, without evidence, more salmon creates a boom and bust cycle. So to answer your question, I would argue freshwater flow and habitat management should accommodate the variability imparted by the ocean and should accommodate what is possible, increasing variability in salmon returns. And that can be done through an ecosystem level model, not simply looking at an average number of salmon returning. So I hope I avoided your question just right enough to make you think you weren't.
That is, it's often an afterthought for drinking water agencies, but certainly clean water agencies have limited resources and often maybe aren't the biggest point source necessarily to improve some of the water conditions. So I'm just trying to prepare to have some of these conversations and give it context. So I think the ecosystems-based model is a good reference point, and a good scale and resolution to try and be a conduit to guide to other needs as far as ecosystem management and the implications of decisions that people have to make while managing urban runoff and customer waste and things like that. So I appreciate your advice. Thank you.
It's also worth mentioning that ecosystem models vary, right? You have models of intermediate complexity where you could have 5 critters in it. But a salmon life cycle model is also an ecosystem model. So we are approaching this from a managerial standpoint, we hope.
Thanks, Michael. Judith, any others on the line?
We have some that were going along with the same topic. We did have one question, one of the acronyms, LMH.
Large marine heat wave in 2015. We had an extremely hot event that altered the ecosystem pretty dramatically. It was referred to as the blob.
Any other questions there? Lynn in Portland, you said?
How much lead time before the ocean is going to respond? Management question?
Gotcha. Okay, I think I'm understanding. What we're asking is, what type of indicator do we have, and how much time do we have from that indicator to be able to change practices within year to improve the return numbers of salmon or whatever our objective is? I am hesitant to apply this in a tactical way to year. And not because I don't think it works super well. I think that when we do those types of approaches, they invariably fail and we lose a lot of confidence in our people. That said, you can use it to some degree to inform, like, the North Pacific High in March. If I know in March the North Pacific High is doing something, that's the same month the PFMC is making their legislations and rules, we can inform the PFMC about what's going on in the system. And we do this through the IEA reports, we do this through the Cal Coffee report, status reports generally. What management practices they can take to meet a poor oceanographic condition in March, other than harvest control rules, I'm not sure. I don't think hatcheries are flexible enough to change their practices within year. So I guess my answer is we would know in March a better understanding of what we can expect to see coming into the salmon fishery and into the seabird breeding populations and into the forage-based communities.
Okay, thank you. Any other questions, either online or in any of the...
Yeah, Alan Merns here in Seattle. I just keep wondering about the conditions that prevailed in the 1920s to 1940s in the California current. We had humongous populations of sardine, maybe hardly any sea lions. I don't know what the whales were doing. I don't know what the salmon were doing. But it seemed like it was a different ecosystem.
Well, I think that you made that ecosystem interesting. You not only said that earlier on maybe we were a cooler ecosystem, and I think those regime shifts are understood. But it was interesting that you brought up the predator populations. And I think while we understand there are these environmental regime shifts, and we saw that with the switch between sardine and anchovy in the late 40s, but now we're coming into a period where we're just not really understanding that we're now into a biological regime shift. We're in a place where we have, and we want them, but we just have to figure out how to manage with them in the context of increasing predators. So I would argue that, yes, the world was a different place then, and now the world is environmentally different, but it's also biologically different. And that brings up questions I often hear. What do we consider to be the ecosystem? What do we want it to look like when we manage it? And if we go back and we say we want this to look like the 20s to the 40s, that might be out of our control, right? So fortunately, with these types of models, you can play. And you can really play with the idea, well, what was it like in the 40s? What will it be like in 2100?
I'd like to see a historical application of the models.
Thank you, Alan. Great suggestion.
Okay.
And thanks to everyone there at the NOAA Library for hosting us from there. And thanks to the West Coast Regional Office here in Portland, Oregon for hosting us here. Our next presentation seminar in the Ecosystem-Based Management Series is on March 14th. And that is going to be Sean Lucy, who is a scientist up at our Northeast Fisheries Science Center. And so please feel free to register for these series on the NOAA Library website, and you'll get a reminder and all the information you need to do to sign in and call in to the next series. Again, these are on the second Wednesday of the month, 3 p.m. on East Coast time, noon on the Pacific time. And again, thanks for everyone's participation and all the great questions. And again, special thanks to Brian for taking all the time out to do this today. So,
thank you.
Thank you.